Good morning students. Today is the lecture 2 of module 1. In today's lecture, we will discuss the classification of membrane processes, and after that we will discuss one by one different membrane processes. Basically, I will give you a basic overview and understanding of different membrane processes. Then we will discuss about their advantages and drawbacks, as well as some major applications of membrane processes in general. As you know, membrane processes are an accepted unit operation for a variety of separation in most of the industries, whether it is a chemical or allied industries. The processes are driven by pressure, concentration or electric force across the membrane and can be differentiated according to the type of driving force, molecular size or type of operations. Now, membrane separation process involves the use of a membrane which is a permeable or semi-permeable phase and restricts the motion of certain species. So, we have understood what is the meaning of permeable or semi-permeable in our last class. So, some of the factors utilized for membrane separation are relative size, electrostatic charge, other non-covalent interactions, diffusivity and shape. In order to drive the solutes and solvents through a membrane, driving force is necessary. If you remember students, last class I told you that in the driving force is of various forms, but the true driving force is always the difference in chemical potential. Since we cannot measure chemical potential directly, hence we have to express the driving force in a measurable quantity or parameter, which can be either transmembrane or hydrostatic pressure, concentration or electrochemical gradient, it can be osmotic pressure gradient, it can be the gradient of electric field, it can be partial pressure or it can be pH also. So, as you can see, all these parameters are different gradients and are readily measurable. So, let us go to the membrane classification. We can classify membranes into various types based upon the nature of membrane, based on application of a membrane, based on structure of membrane and based on mechanism. So, according to this particular slide, you can see that based on nature of the membrane, it can be natural or synthetic. Based on application, we can have gas, liquid, liquid. Based on structure, we can have either porous or non-porous membrane, the dense membrane, or based on mechanism, it can be either convective, adsorptive, diffusive or ion exchange. Now, though the earlier slide gave a general classification, in a better way we can classify the membrane processes as given below. So, this I have tried to classify membrane processes according to these 5 types. So, first one is based on material, second one is based on membrane structure and morphology, third one is based on operational modes, fourth one is based on charges and fifth one is based on driving forces. So, we will see one by one. So, the first one is based on materials. You can see that either we can have a natural polymer, which is cellulose is an classic example of a natural polymer. So, cellulose is one of the most abundant only available natural material. Then otherwise we can have synthetic polymers. So, most of the commercial membranes which are available today in the market are all synthetic membranes. So, it can be prepared either from polysulfone or PVDF which is polyvinylidene fluoride, or polyether sulfone which is a modified version of polysulfone, or polyacrylonitrile or even nylon. So, apart from the natural or synthetic polymers, we can have inorganic membranes also, which are usually called as ceramic membranes. So, these membranes can be prepared from various materials. I have just listed 3. These are common materials, either alumina, titania or zirconia. So, next classification is based on structure and morphology. So, under this we can either have a symmetric membrane or asymmetric membrane or a composite membrane. Now, symmetric membrane is one in which the pores can either form long channels or the membrane can have a porous sponge type structure. Usually the thickness is 10 to 100, 200 micron, and a decrease in membrane thickness results in an increased permeation rate. The less the membrane thickness, the less will be the resistance provided by this membrane, so that the more will be the permeation and the more will be the flux also. So, asymmetric membrane is one in which the membrane is constituted of 2 or more structural planes of non-identical morphologies. So, 2 different types of morphologies are fused together to give a asymmetric membrane, and composite membrane is one which have chemically or structurally distinct layers. So, nowadays we have usually the ceramic composite membranes in which ceramic part provides the porous or the base support, over which we can have a polymeric skin layer either in microfiltration or ultrafiltration range to achieve a desired separation. So, you can see how it looks like basically, though we have covered this little in our first class. So, symmetric membrane, either we can have cylindrical pores. Usually, as I told you earlier also, cylindrical pores are very difficult to achieve, only the zeolite membranes have the cylindrical pores. So, most of the membranes will have different types of pore sizes and shape. And we can have a dense or non-porous membrane also. Under asymmetric membrane, we can have 3 different types of membrane. The first is a porous membrane with a porous top layer, the first one you can see. So, we can have a porous membrane with a porous top layer, and this you can see the porous, this is also porous, the here support is also porous. So, another one, we have a porous support here basically with a non-porous top layer. So, this layer is actually a non-porous layer. So, this is another type of asymmetric membrane, and the last one is a composite membrane, and in which there is a top non-porous membrane, okay, here this one, and we can have a porous support here, and intermediate also we can have another membrane with a little less porosity, pore size than the below support one. So, we can prepare it basically in a 2-step or 3-step preparation. So, the next one is based on operational mode. So, we can have a dead end filtration or we can have a cross flow filtration. So, mostly the membranes works in these 2 different modes. So, a dead end filtration, I will just try to draw something. So, here you can see. So, let us say this is a cell in which I have put the membrane here. So, this is my membrane, okay, and then we can fill in my mixture here which containing the solute which I want to separate, and then I pressurize the system. I applied pressure. So, I will get permeate here. So, this is my delta P that I am pressurizing, this is my feed, okay, and this is what I will get is permeate. Okay, whatever is will be retained on the surface will be retented. So, this is a simplest form of our dead end filtration, and in which you can see the permeate flux declines, just like here the permeate flux, initial permeate flux will be very high and slowly the permeate flux will decline. Flux decline will be more in dead end filtration mode. So, then the next one is the cross flow filtration. In cross flow filtration, the flow is you can see this is actually feed is coming from here, okay, and it is passing over the membrane surface, okay, and we get retented here and we get permeate here. So, one of the best thing in cross flow system is that since the flow is perpendicular to our this one membrane surface, okay. So, the deposition whatever the deposition is happening here on the surface of the membrane which causes the concentration polarization will be little less than dead end filtration mode. And of course, flux decline profile will be same as in the case of dead end filtration. However, the flux declining rate will be less than that of the dead end filtration. So, the next one is based on charges. So, we can have 3 different types of membranes here. One is anion exchange membrane, another is cation exchange membrane, and then the next one is bipolar membrane. Now, what is anion exchange membrane? So, the membranes containing fixed cationic charges and mobile anions that can be exchanged with other anions present in an external fluid in contact with membrane. So, similarly, in a cation exchange membrane, the membrane will be having fixed anionic charges. So, please see this figure in which you can see then in an anion exchange membrane we have fixed cationic charges. So, these are the fixed cationic charges, these are fixed inside a anion exchange membrane. So, anion exchange membrane basically means this membrane will have fixed cations and this membrane will exchange anions. Similarly, in a cation exchange membrane we have fixed cations, okay, and this will exchange the anions. So, cation exchange membrane is one in which cations will exchange with the help of fixed anions which are imparted inside the membrane. Then we can have a bipolar membrane also. So, it is again a synthetic membrane containing 2 oppositely charged ion exchange layers in contact with each other. So, we can have a cationic layer and we can have a anionic layer fused together, okay, that will do the separation for some specific purposes. So, the next one is based on driving force. So, we can have 3 different types of classification under this particular sub classification. We can have pressure driven membrane, under which the microfiltration, ultrafiltration, reverse osmosis, nanofiltration comes. Then we can have a concentration driven system or a membrane, under which dialysis, hemodialysis comes. Then we can have a potential driven membrane, which is electrodialysis and electroosmosis, basically under the influence of a electric gradient. So, membrane process classification, apart from these classifications, we just try to understand how membranes can be classified. Membrane processes can be classified into various sub classifications. So, there are other membrane processes such as facilitated or carrier mediated membrane processes, liquid membrane based separation, pertraction or perstraction also, piezo-dialysis, membrane reactors and bioreactors. Now, to add on, there are hybrid separation processes where more than one unit operations are combined together to achieve a targeted separation, and in a membrane hybrid system, membrane is one part and another part can be either distillation, absorption, adsorption or any other such unit operation. So, one of the membrane processes will be the inherent part of the hybrid process, and there can be any other processes which is joined together joined with the membrane process to give us a particular yield or separation. So, one classic example of this hybrid process is the hybrid distillation and pervaporation process to separate a 50-50 azeotropic mixture. So, apart from this, there are many processes which are still under development. So, the principal characteristics of various commercialized membrane separation processes can be specified on the following 7 aspects. These are very important. So, these 7 aspects are separation goal, the nature of species retained, nature of the species transported through the membrane either electrolytic or volatile, minor or major species of feed solution transported through membrane, the driving force, mechanism of transport and selectivity, phase of feed and permeate streams. So, these are the 7 principal characteristics for the commercial membrane separation. So, let us now see, this slide will tell you how the membranes which basically separate based on the size. So, I have given the 4 basic membrane pressure driven membrane processes: microfiltration, ultrafiltration, nanofiltration and reverse osmosis. Please have a close look to the slide. You can see that in microfiltration, water, then different monovalent ions, multivalent ions as well as virus will pass through. That means, the microfiltration membrane will allow all these to pass through the permeate side, whereas it will retain bacteria and suspended solids. Then the ultrafiltration, all these will pass through, however, it will reject viruses, bacteria as well as suspended solids. In nanofiltration, again some of the monovalent ions depending upon the sizes will be retained and some will pass through, and it will reject virus, bacteria and suspended solids. Okay. And in reverse osmosis, only water or solvent can pass through, all other ions, viruses, bacteria, any suspended solid will be retained. So, what we understand from this particular slide is that as we move from microfiltration to ultrafiltration, then nano to reverse osmosis, the pore size of the membranes decreases. Since the pore size of the membrane decreases, all these processes will need more delta P. That means, the more transmembrane pressure is required, so that it will separate, it will give a desired separation. Now, we will see one by one different membrane processes, and I will try to give a brief introduction about the different types of membrane processes, starting with microfiltration. So, as you know that microfiltration can be defined as a pressure driven process where it is used to separate micron sized particles from fluid. There are 2 types of microfiltration configuration, namely one is dead end and cross flow filtration. We just saw under the classification when I show you the dead end filtration mode and a cross flow mode, and the dead end filtration is suitable for dealing with suspensions with very low solid content. And please note that the dead end filtration is not a commercialized industrial application. This is used only in laboratory scale to characterize certain membranes and to develop a process. there is no industrial applications of that, and the major application is of course the cross flow filtration. It is used for higher concentration as the deposits on the membrane are swept away by parallel flow on membrane surface. So, a very highly concentrated solvents or mixtures can be processed through cross flow filtration, since the depositions are depositions of the solutes on the surface of the membrane are usually swept away by the parallel flow on the membrane surface. So, transmembrane pressure ranging for microfiltration is usually 1 to 50 psi. So, that is the driving force. If pore size of the membrane is smaller than the particles in the solution, surface filtration will result. This means, if the particles are higher than the particle size is basically higher than the pore size of the membrane, then all of them will be retained on the surface of the membrane. So, all of them will be rejected. Otherwise, depth filtration results as particles are caught in membrane's interior. Otherwise, what will happen if the particle sizes are solute sizes are smaller than the membrane pore size. So, these particles or solutes will pass through the pores of the membranes. So, some of them will be passing through and then it will go through the permeate. Some due to the various shapes and sizes of the pores will be retained in the inside the membrane pores, thereby creating internal fouling. So, some application of microfiltration includes cell harvesting from bioreactors. When we culture various cells, bacteria, algae or all these things, so they need to be harvested once they are mature enough. So, microfiltration will help us in this in achieving this target. Then water purification, then removal of cell from fermentation media. Basically, it is removal of the dead cells, then air filtration and industrial wastewater treatment. So, next is ultrafiltration. So, ultrafiltration is used to separate large molecules of solvents. The molecule size are usually larger than that of the microfiltration, and separate large molecule from smaller molecule, that basically it is fractionation, and large molecules from other molecules. So, the primary mechanism is size exclusion, just like our microfiltration, but chemical interactions between solute and membrane and other operating parameters can also affect the process. So, as I told you in the last class that one of the two very most important things which should be noted for any membrane separation process. First one is the physicochemical properties of the solute that I want to separate. Then the second one is the physicochemical properties of the membrane, or the basically we can say the membrane material. Then normal transmembrane pressure for the ultrafiltration ranges from 10 to 100 psi. So, ultrafiltration is widely used for concentration of solutes by removal of by removing the solvent, purification of solvent by removal of solute, fractionation of solutes, either binary fractionation, tertiary or even more than that. So, we can have cascades also. Then diafiltration, that is removal of salt and other low molecular weight compounds from solutions of macromolecules. You can see this figure. Okay, the ultrafiltration membrane is allowing the passage of water, salts and dissolved organics, whereas it is retaining the bacteria, macromolecular organics as well as particles and silts. Then we will discuss about nanofiltration. So, nanofiltration is almost similar to that of reverse osmosis. However, membranes are slightly more permeable. Since the membranes of nanofiltration are little more permeable, hence the pressure required for nanofiltration is obviously lesser than that of the reverse osmosis. And nanofiltration separates at molecular level, removing all suspended solids and most of the dissolved solids. So, nanofiltration spans the gap in particle size between reverse osmosis and ultrafiltration. So, basically nanofiltration lies between somewhere between reverse osmosis and ultrafiltration, and that is why it is also called as ultra osmosis. So, the transmembrane pressure for this nanofiltration usually ranges from 40 to 200 psi. So, the applications of nanofiltration includes seawater desalination, then concentrate sugar and clarify sugar stream in sugar industry. It has huge application in sugar industries. Then deacidification of whey during the cheese production, and recovery of dyes in textile industries, separation of heavy metals from water and wastewater, removal of sulphate, phosphate, nitrate and fluoride. If you just look at the figure below, you can see that nanofiltration will allow membrane will allow only water and salts to pass through, whereas all other dissolved organics, bacteria, viruses, any other macromolecular organics and particles and silt will be retained or rejected by it. Then the next membrane process is called reverse osmosis. So, all of you must have studied about osmosis during your school times. So, osmosis is a natural phenomena. So, it occurs when the water passes through a membrane from a site with lower solute concentration to a higher solute concentration until the osmotic equilibrium is reached. So, this is what is actually the water, that is solvent, is passing from here to here, okay, using this is the membrane. So, till osmotic equilibrium reached, okay, the flow will takes place. So, this is the osmotic pressure we can see from here, okay, the concentrated solution side. So, and the membrane is of course a semi permeable membrane which will allow only the solvent or water to pass through and retain all sorts of ions. So, to reverse the water flow, to reverse this particular flow, what is required is that we need to apply a pressure, okay, mechanical pressure, okay, which will overcome this osmotic pressure, whatever it is there, okay, the delta pi, right. So, we apply a external pressure here, right. So, this pressure will try to overcome this osmotic pressure, okay. So, as a result the separation of water from a solution becomes possible, okay. This phenomenon is called reverse osmosis. So, that means what we are doing, we are acting against the gradient of natural chemical potential, right. So, usually the flow should takes place from here to here during osmosis. Since we want to remove solvent from the concentrated solution, usually the application is desalination of sea water, in which the sea water which is highly concentrated with salt has to be desalinated, okay. So, in that case we will use reverse osmosis. We apply the pressure which will overcome that osmotic pressure, so that your flow will starts from, okay, this side to this side. So, reverse osmosis process allows selective passage of a particular species, usually the solvent, while other species solutes are retained or partially or completely. Solute separation and solvent permeability are membrane characteristics and are dependent on the membrane material and structure of the membrane layer. So, application of transmembrane pressure to concentrated solutions forces solvent through the RO membrane towards the lower concentration. So, few important applications of reverse osmosis are sea water desalination. So, which is the most important application of reverse osmosis, then wastewater treatment, even water treatment also, and ultra pure water production. So, you can see from the figure is that all these things, algae, plankton, suspended solids, pathogens, macromolecules, small molecules, salts, everything will be rejected by the RO membrane, and only solvent will flow through the RO membrane. Okay. So, the next process is dialysis. So, dialysis is a membrane separation process in which one or more dissolved species flows across a selective barrier in response to a difference in concentration. So, please remember that dialysis is a concentration driven membrane separation process. So, the mode of transport is usually diffusion, and the separation occurs because small molecules diffuses more rapidly than larger ones. So, it is quite understood that smaller molecules diffusivity will be higher than the larger molecules. So, the rate of diffusion plays a very important role in separating of these molecules. So, you can see this particular figure, you can see that through this membrane, through our this dialysis membrane, sodium, potassium, calcium can pass through. Okay. Whereas creatinine and urea, which are large molecular components, will be rejected by the dialysis membrane. So, this is what is the application of basically hemodialysis is, where the blood is purified. So, the application of dialysis includes the removal of acid and alkali from products, removal of alcohol from beer. Nowadays, there is a lot of demand of making alcohol free beer in western countries. So, that is why alcohol will be removed from the beer using this dialysis membranes, removal of salt and other low molecular weight compounds from solution of macromolecules, then concentration of macromolecules as well as hemodialysis. So, one of the most prevalent application of dialysis is the hemodialysis. So, the next application is gas permeation. Gas permeation or gas separation is one of the most important application of membrane separations, and it has lot many industrial applications. So, gas mixtures can be separated by either porous or dense membranes. We can use both porous and non-porous membranes. So, in general, the transport through membrane is based on Nodson diffusion, solution diffusion or molecular sieving. So, I have just tried to show you 3 different types of mechanism through which the gas separation takes place. You can see this is the membrane, okay, and this is the upstream side and this is the downstream side of the membrane. In 3 cases, the first one is Nodson flow. So, here the separation is based on the inverse square root of ratio of the molecular weight of A and B, if I am separating both A and B. Okay. So, Rp is the diameter of the pore, this is one pore and Rp is the diameter of the pore. So, the separation is happening due to Nodson diffusion. The next one is molecular sieving, in which the separation is based on diffusion and sorption characteristics, right. So, the rate of diffusivity of the solutes plays an important role in this type of separation. The next one is solution diffusion separation, as you understand the name solution diffusion. So, the separation is based on both the solubility as well as diffusivity of the solutes for the separation. Okay. So, gas separation has lot many applications. So, the one of the most important commercial application is the hydrogen recovery from synthesis gas. Okay. Then oxygen enriched air for combustion and medical applications, nitrogen enrichment for wafer fabrication, metallurgy and other applications, carbon dioxide for enhanced oil recovery, removal of carbon dioxide, then helium for natural gas. Now, you can see this particular application in which we are trying to remove carbon dioxide from a mixture of methane and carbon dioxide. So, the feed contains both methane and carbon dioxide, and I use a gas separation membrane, or I can call it as a carbon dioxide selective membrane, in which the carbon dioxide will be selectively separated or permeated through. So, we get more carbon dioxide here and we will get purified methane here. So, of course, due to the porous or inherent nature of the membrane, some methane will come to the membrane side. Similarly, so carbon dioxide, which is very trace, will also go through the methane. But we will get we are having nowadays we have excellent carbon dioxide selective membranes. So, which will which will permeate carbon dioxide almost up to 95-98% or even more than 99% also. So, another application is little this one extended application of gas absorption is gas absorption and stripping. So, unlike the conventional membrane process, here the membrane is the selective barrier or selective layer towards the fluids to be separated. The membrane used in absorption or stripping processes act only as somewhat a packing material as shown in the figure. So, you can see this instead of having a packed bed for doing the adsorption, we can have membranes, okay, packed as a packing material. So, we can have the absorbent in here, okay, and absorbent out here. So, the same application of the carbon dioxide and methane, which is present as the feed, okay, will be used here, okay, to separate or to get purified methane. So, carbon dioxide will come out here. So, the advantages and disadvantages of using membranes in gas absorption are it provides large surface area per unit volume. Okay, that is very important, because to have a better mass transfer we should have more surface area. So, in a smaller place we can have more surface area by providing this type of membranes with reference to, of course, the conventional absorption system, and no flow constants such as flooding or loading exist as those exist in usual conventional absorption processes, and membranes supply new mass transfer resistances, that of the membrane, which is not present in the packed column. So, this is one of the disadvantages of having membranes as a packing materials. So, the membrane thickness will provide a resistance to mass transfer. However, if we can have a very thinner membrane, then we can almost nullify the resistance to flow, which is being provided by the thickness of the membrane. The next is pervaporation. one of the most important application in membrane separation. So, it is a process where a liquid mixture is in direct contact with one side of the membrane and where the permeate stream is removed in vapor state. So, this is very important, the permeate is getting removed in a vapor state, okay, from the other side of the membrane. So, the pervaporation, the name comes actually from what I said just before, that it is permeate vaporization. The permeate is getting vaporized. So, pervaporation separates usually a low volatile or low boiling point liquid from a non volatile liquid, and solution diffusion is the mechanism of the separation. Please have a look at this figure. You can see the feed is entering here. This is your membrane, right, and what is happening, the feed is liquid and whatever retentate I am getting, that is also a liquid, right. The permeate, when it is passing through the membrane here, okay, is getting vaporized. So, I can achieve the vaporization of the permeate by two processes. Either I can have a vacuum pump here, which will create the vaporization of the permeate here. So, thereby creating, basically it will create a low vapor pressure there, and otherwise I can have a sweeping gas. So, either I can have a sweeping gas somewhere here, I can pass a sweeping gas so that it will pass through the, so it will create a low vapor pressure, okay. And the condenser is optional. If we want the this one permeate as a liquid phase, then we can have a condenser. So, the major applications of pervaporation are the breaking of azeotropic mixtures or the separation of azeotropic mixtures. As you know that usually the azeotropic mixtures can be separated by azeotropic distillation, or we can go extractive distillation, where we are adding a third solvent, thereby increasing the cost of the entire process. Then we can have the heat sensitive product separation using pervaporation. Dehydration of alcohol is a classic example, and it is commercialized process also, and then removal of VOC, volatile organic compounds, from contaminated water. So, these are some of the classic examples of membrane pervaporation process. So, the next process that we will discuss is electrodialysis. So, it is a dialysis process which is happening under a electric gradient. So, electrodialysis is a electrochemical process used to separate charged particles from aqueous solutions or from neutral solutes. So, it is the movement of ions through ion selective membranes under the influence of an electromotive force applied across the membrane area. So, positively charged cations such as sodium and calcium migrates to the cathode, whereas negatively charged anions such as chloride and sulfates migrate to the anode. So, please see this particular figure or the schematic here. You can see there is a anode here, there is a cathode here, and we have here in this particular figure we can see 4 different types of membrane. So, the red one is cation exchange membranes, and the violet color one is the anion exchange membrane. So, a cation, as we understood previously in this lecture only, the cation exchange membrane will have fixed anions and anion exchange membranes will have fixed cations. So, feed is passed from the below, I can pass the feed from the top also, then I got dilute from here and concentrate from here. So, the applications of electrodialysis process are desalination of sea and brackish water. This is one of the most important applications. So, where desalination can be achieved by electrodialysis as well as even by reverse osmosis or nano filtration also. So, purification of organic compounds, wastewater treatment, wine stabilization, where we remove actually the tartaric acid which is forming when the fermentation is happening, and then recycling and regeneration of galvanic baths in the metallurgical industries actually. So, the next process is actually liquid membranes. Students, I told you in the next class what is actually basically a liquid membrane. A liquid membrane is essentially the membrane is made up of a liquid. So, here the liquids such as hydrophobic solvents form selective barriers between aqueous feed solution and aqueous absorption phase. So, please see look at the figure. So, we can have 3 different types of liquid membranes, BLM, SLM and ELM. So, the BLM stands for the bulk liquid membrane, SLM stands for the supported liquid membrane and ELM stands for the emulsion liquid membrane. So, bulk liquid membranes are not commercialized, they are utilized in the lab scale to generate data and to see whether the process of liquid membrane is feasible for a particular separation or not. And in a SLM, SLMs and ELMs are already adapted in various industrial sectors. So, SLM is a supported liquid membrane where there is a support. So, this you can see this is a membrane. So, this can be a microporous membrane or microfiltration membrane, and what we are doing is basically we are putting the membrane phase which is present in the pores, inside the pores. So, this is a particular pore. So, you can see that the membrane which is a liquid is impregnated inside the pores of the membranes. So, the separation will be achieved here only. The membrane itself is not doing anything, it is just providing the mechanical support or it is just holding the liquid membrane phase. Similarly, in the case of emulsions membrane, so the membrane phase you can see that this is the membrane phase, E is the membrane phase, this is the feed phase, okay. R is the receiving phase. Okay. So, the membrane phase is will do the separation of the solutes from the feed. So, the separation will happen like this, then eventually the solute will go to the receiving phase and we will get inside the receiving phase. Then we can have emulsifier breakers which will break the emulsifier, we will get the solutes we want to separate from the feed stream, that will go to the receiving phase and then we can recycle the membrane phase again. The major advantage actually of the ELM is that we can create very high membrane specific surface area, almost 1000 to 3000 meters cube per meter cube, okay, in a very small space. Okay, the application of liquid membrane includes removal of heavy metals from effluents, dephenolation of wastewater, that means removing phenol from various water and wastewater systems bodies, then separation and concentration of amino acids, enzymatic bioconversions and gas separation. So, the next membrane method is membrane reactors. We will just go through a little basics of what is membrane reactors. Later on we will discuss in one of our lecture in detail about membrane reactors and bioreactors. So, the membrane reactors combine reaction with separation to increase conversion. The main feature of the reactor is to remove the reaction product out of the reactor using the membrane, so that the equilibrium of the reversible reaction is shifted and the reaction continues to proceed toward completion. So, I am continuously removing one of the reaction product which is not a value added product for me, so that the main product yield will be higher than that of the byproduct. So, the three general types of membrane reactors are possible. One, so the first one is membrane act as a contactor. So, basically here in this type of system, the job of the membrane is to separate catalyst from the reaction medium. Catalyst means the solid catalyst, the heterogeneous catalyst. Then membrane act as a separating barrier. Here the job of the membrane is to shift the equilibrium of a chemical reaction by selectively removing the reaction products. One of the reaction products or a bit more than one of the reaction products. And the another one configuration, membrane act as both a contacting and separating barrier. So, let us see this figure. Here you can see there is a mixture of A and B. So, this is A and this red one is B. So, both are passing or entering through the membrane reactor, and inside the membrane reactor catalytic conversion takes place. So, we can see two ceramic membranes here, tubular ceramic membranes here. There can be many more, just for our understanding we have seen two, I have shown two here. So, C is the product which is forming inside the membrane reactor and then it is coming out here. So, we are getting the product here, and what these membrane reactors are doing, they are selectively removing the B. This is B, they are selectively removing the B, so that the yield of C will be more. So, C is produced in the reactor and B is diffusing through the membrane pores. So, B is selectively removed, so that there will be no B present here in the product, and yield of the product which is C will be more. And now, let us see the different advantages and disadvantages of the various membrane processes. So, The first and foremost important advantage of the membrane process is appreciable energy savings. I told you in the last class also in a nutshell, but let us see what is the meaning of that. Actually, compared to conventional processes, membrane separation technology permits concentration and separation without the use of heat. In all other conventional processes, we need heat to do either concentration or separation. Here we do not need. So, there is a lot of energy saving here. So, evaporation and freeze concentration are common dewatering techniques used for liquid products. Evaporator requires input of almost 100 BTU per pound, while freezing requires 144 BTU per pound to change the state of water from liquid to vapor and liquid to solid respectively. Since membrane process do not require a change in the state of the solvents, so they do not require a change in the state of the solvent except maybe pervaporation. So, to effect dewatering, it resulted in considerable saving in energy. Then clean technology with operational ease. So, the membrane operations, no complicated heat or heat transfer or heat generating equipment is required. These processes could be operated at ambient temperature. So, the next one is greater flexibility in designing systems and hybrid process development. As I told you earlier also, one of the best advantage of membrane process is that it can be hybridized along with any other unit operation such as distillation, absorption, adsorption and any other such operations. So, membrane unit operations are uses as part of the process design with other unit operations. For example, dehydration of organic solvents is done mostly by distillation. Membrane modules that can selectively separate water from many organic liquids including alcohols are available. It is more advantageous to use both distillation and membrane unit operations in series. So, if you do a combination of distillation and membrane, okay, a hybrid system, then we can save appreciable energy and even space also. The hybrid system can successfully increase the capacity of existing plants and improve the product quality. A hybrid process is successful if its overall cost and performance is an improvement over the individual alternatives. Otherwise, why should we go for a hybrid system? So, next is disadvantages of membrane system. So, we understood what are the advantages. It is not always that membrane is win-win situation for any prevalent separation need. There are certain disadvantages. The first one is membrane fouling. So, membrane fouling is a pertinent problem in membrane separation process. The contaminated feed increases the rate of membrane fouling, especially for hollow fiber modules. So, as you understand, the last class actually we discussed how fouling taking place. So, fouling is a consequence of concentration polarization. So, concentration polarization is the deposition of solutes over the membrane surface which happens gradually. So, when excess deposition happens, some of these solutes will pass through will try to squeeze themselves and get inside the pores of the membrane, thereby blocking the pores mouth as well as pores the constricted passage of the pore inside the membrane. So, thereby doing two types of fouling, either external fouling or internal fouling. Then the next one is upper solid limits. So, membrane processes are limited in their upper solid limits. So, what is the meaning of this? Now, in RO reverse osmosis, it is the osmotic pressure of the concentrated solutes that limits the process. Okay. So, you have to in RO you have to just cross the barrier of the osmotic pressure, then only by providing a mechanical pressure excess than that of the osmotic pressure, so that the separation will happen. So, in case of ultrafiltration and microfiltration, the low mass transfer rate and high viscosity makes the pumping of the retentate difficult. So, if you are having a very highly viscous solvent which is our retentate, then taking it out, so basically pumping it out from the membrane module or the membrane reactor system becomes bit difficult and challenging. Then the next one is expensive. So, compared to other processes, membrane processes are expensive due to the cost involved in fabrication, occasional replacement, fouling and poor cleanability of the membrane. But nowadays with huge advancement in membrane science and technology, the cost of the commercial membranes have come down drastically. So, the major areas of membrane applications. So, let us see a few areas and we will try to understand how membrane plays a role in few of the industrial applications. So, membrane processes are no longer bound in the domains of the laboratory, but are found in extensive applications in various industries. So, the processes have achieved impressive industrial importance for the resolution of aqueous liquid mixtures, purification of chemicals and biological products, wastewater reclamation and many more such applications. However, membrane technology is not a panacea for all recovery and separation process. In many cases, the other processes are either more effective or less expensive, or it can be both. So, you can see the different industrial sectors and the applications. So, in the drinking water we can use either ultrafiltration, nanofiltration, reverse osmosis. In demineralization of water we can use RO, electrodialysis and wastewater treatment, whether it is direct physical treatment or membrane bioreactor. We can use all sorts of membrane pressure driven membrane processes as well as electrical driven membrane processes such as microfiltration, allofiltration, reverse osmosis, electrodialysis. And in food industry we have a major application in food industry, the membrane application, whether it is dairy industries, meat industries, fruits vegetable processing industries, sugar industry. We have use ultrafiltration, RO and ED membranes, electrodialysis systems to do the separation. In beverages industry like fruits and vegetable juices, wine and breweries, tea and coffee industry also use microfiltration, ultrafiltration, RO and pervaporation membranes. In biotechnology industries, the applications of ultrafiltration and microfiltration is more prevalent than any other membrane processes. So, in chemical industries basically the gas separation, where whether it is we talk about the hydrogen recovery, carbon dioxide separation and nitrogen enrichment, we use the gas separation membranes, specific gas separation membranes to do the separation. And when we go for a vapor liquid separation, just like our for example ethanol dehydration or organic recovery, so we use the pervaporation membranes. So, in case of fuel cells, we use proton exchange membranes which PEM which is called PEM fuel cells. Okay. So, in chemical industries the membrane separation process has emerged as the most important and practically useful membrane separation technique. So, some of the major applications are production of process water for industrial use, wastewater treatment, desalting and demineralization of food, acids and reactive dyes, concentration of different types of dyes. So, besides this, membranes have also established potential use in energy generation by proton exchange membrane fuel cell, greenhouse gas separation and other such technologies. So, then next is the pharmaceutical industry. In pharmaceutical, or many times it is called as biopharmaceutical industries, membrane has a lot many applications. So, due to the macromolecular nature of most bioengineered products along with the harvesting and fermentation technologies, so membrane like ultrafiltration and microfiltration plays a vital role in doing the separation. So, membrane technology plays a key role in following applications. So, concentration and purification of soluble macromolecules such as plasma protein, vaccines, enzyme, yeast and even the antibodies, process water as per standards, because ultra pure water is required for biotechnological applications and in pharmaceutical industries, and endotoxin free waters. In food and dairy industries, the lactose and protein concentration we use usually either microfiltration or ultrafiltration membranes. The concentration of whole and skimmed milks we use microfiltration membranes. Lactose protein separation, ultrafiltration and microfiltration both can be used. So, fractionation and concentration of egg albumin, fish oils, proteins, concentrations extracts from vanilla, lemon peel, malt, etc. can be achieved by either by using ultrafiltration or even pervaporation membranes. So, let us see this is one classical example of a schematic applications of various membrane processes in milk production or milk processing industries or dairy industry basically. You can see the milk first goes to the microfiltration unit here, where it removes the bacteria and microorganisms, then it goes to another microfiltration units where the fats will be removed. So, we will get basically skimmed milk, then again it processed through a ultrafiltration unit where we will get condensed milk or cheese, and then it passes through a reverse osmosis membrane where we get water here and lactose and mineral salts here. So, the stream from here it can go to a electrodialysis unit where the product is desalted whey and we get mineral acids here. Similarly, we can use ultrafiltration and microfiltration to separate proteins, peptides and as well as other fermentation products. So, this schematic representation makes us understand that how different membrane processes such as ultrafiltration, microfiltration, RO and ED plays an important and vital role in milk processing or dairy industries. So, the next application is biotechnology industries. So, two major applications of membrane technology in biotechnology are the separation and purification of biochemical products, which are often known as the downstream processing. So, in most of these industries, the downstream processing part, which means the purification or removal of the product from the fermentation broth or the solvent in which they are present is called downstream processing. And that cost is almost 40 to 50% of the entire cost of the product. So, membrane processes such as microfiltration, ultrafiltration, nanofiltration are used for harvesting, concentration and purification processes. Some major applications are enzyme concentration, fermentation, broth clarification, separation of microsolids like antibiotics and vitamins, purification and concentration of vitamins, tissue culture reactor system and bioremediation. So, bioremediation is again a wastewater treatment process in which we remove or remediate various organic compounds present, organic and toxic compounds present using various microorganisms. So, the next slide will tells us about the application of membrane technology in bioprocessing industries. You can see that this is a fermenter which is doing the fermentation, okay, to which we will use actually media and nutrients, and we need water for that. And you can see there is a RO process here. So, water, the tap water which is coming from the tap is being fed to the RO system and it will give us a pure water which is being fed to the fermenter. And then air is oxygen is required for the fermentation of the microorganisms. So, oxygen is being purified using a gas separation membrane from the ambient air. So, now once the fermentation is happening, so we need to remove some of the cells. So, these cells can be either dead cells or the mature cells. So, we remove the cells using microfiltration. Some of the cells can be recycled back, okay, to the fermenter to maintain a proper biomass, and some of the cells will go to the harvesting for the product. Then from here we can place another membrane separation unit, okay, and it will be fed to the chromatographic system where we can get the purified and concentration product. Please understand that chromatography is one of the most important and the only important process which will give us more than 99.99% pure product. This is the only process which will give us such a purity. No other process, even if it is membrane also, will not give us so much of pure product. So, the finishing stage is in most of the biotechnology or biopharmaceutical industries is always the chromatographic separation. So, we understand that how various membrane processes, whether it is gas separation, whether it is reverse osmosis, whether it is microfiltration is being utilized in a bioprocessing industries to purify products as well as remove some contaminants from the product stream. So, with this we come to the end of this particular lecture today. So, students, these are some of the textbooks and references book from which I have taken the materials. Mulder, that is the most important book which I am referring, Basic Principles of Membrane Technology, B.K. Datta's Mass Transfer and Separation Processes, Professor Kaushik Nath's book on membrane separation processes, this is by Prentice Hall, India. Then Cherian's books on ultrafiltration and microfiltration handbook, and Richard Becker's book on membrane technology in applications. So, you can refer these books for more detailed understanding of the processes. So, thank you very much, and in the next class we will cover this following syllabus. So, we will try to understand that 3 or 4 different types of materials which will be utilized to prepare membranes. So, we will be discussing membrane materials in the next 1 or 2 classes. So, we will discuss about cellulose acetate, polyamide, polysulfone, polyether sulfone, all these membrane materials, their properties and their advantages and disadvantages. So, thank you very much. You can write to me at kmohanty@iitg.ac.in if you have any query, and I will definitely try to reply you within a stipulated time. Thank you very much.